Assalamualaikum warahmatullahi wabarakatuh. We are from group 7 and today we're going to talk about cell structures. Before we do that, we're going to introduce our group member first. First, we have Adrian, Aulia, and Salwa. So what we're going to talk about is the first we have detailed cell structures, cell membrane, protoplasm, cytoplasm, organelles, and cytosol. Next, we have structures and functions of the organelle. Next, we have Endosymbiont Theory. And last, we have a Prokaryote Structure.
Point number one, detailed cell structure. Number one, protoplasm. Protoplasm consists of all the living material inside the cell. Number two, cytoplasm. The cytoplasm is all the content inside the cell excluding the nucleus. Next. Number three, organelle. An organelle can be defended as a functionally and structurally distinct part of a cell. Number four, cytosol. The cytosol is fluid that is present in the cell membrane.
Next, we have the cell structures. First, we have cell membrane. It is a double membrane. It's composed of lipids and proteins present in both plant and animal cells. Its function is to provide the shape, protects the inner organelles of the cell, and acts as a selective permeable membrane. Next, we have chloroplast. It's present only in plant cells and contains a green-colored pigment known as chlorophylls. Its function is to be the sites of photosynthesis. Next, we have cytoplasm. It's a jelly-like substance consists of water, dissolved nutrients, and waste products of the cell. Its function is to be responsible of the cell and metabolic activities. Next, we have Golgi apparatus. It's a membrane bond sac-like organelles are present within the cytoplasm of the eukaryotic cells. Its functions, it's mainly involved in secretions and intracellular transport. Next, we have endoplasmic reticulum. Endoplasmic reticulum is basically divided into two, which has the soft endoplasmic reticulum and the rough endoplasmic reticulum. But basically, endoplasmic reticulum has a structure of network of membranous tubules is present within the cytoplasm of a cell. Its function is to form the skeletal framework of the cell involved in detoxifying and producing lipids and proteins. Next, we have lysosomes. It's a tiny circular-shaped single membrane bond organelles filled with digestive enzymes. Its function is to help with digestion and removes waste and digest dead and damaged cells. Therefore, it is also called the suicidal bags. Next, we have mitochondria. It's an oval-shaped membrane bond organelle, also called as the powerhouse of the cell. Its function is to be the main site of cellular respirations and also involved in storing energy in form of ATP molecules. Next, we have nucleus. It is the largest part of the cell. It has a double membrane bond organelles, which contain all the cell's genetic information. Its function is to control the activities of the cell and helps with cell divisions and control the heredity characteristics. Next, we have peroxisomes. A membrane bond cellular organelles is present within the cytoplasm, which contain the reducing enzymes. Its function is to involve the metabolic of lipids and catabolism of long-chain fatty acids. Next, we have the plastids. It has a structure of doubled membrane bond organelles. Its function is to help in the process of photosynthesis and pollinations, impart color to leaves, flowers, and fruits, and store starch, proteins, and fats. Next, we have ribosomes. It is a non-membrane organelles are found floating freely in the cell cytoplasm or embedded within the endoplasmic reticulum. Its function is to be involved in the synthesis of proteins. Next, we have vacuoles, a membrane bond fluid-filled organelles found within the cytoplasm. Its function is to provide shape, rigidity to the plant cell and help with digestion, excretion, and stores of substances. Next, we have cell wall. The cell wall is only found in plants. It is also made out of cellulose. It's rigid and it's on the outermost part of the cell. Its functions is to give the cell some shape and to protect the cell from environmental influence. Last, we have cilia and flagella. Cilia are are hair-like projections, small structures present outside the cell wall and work like oars to either move the cell or extracellular fluids, while flagella are slightly bigger and are responsible of the cell movement.
Point number 3, endosymbiont theory. How eukaryotic cells could have evolved from prokaryotic cells that live in symbiosis. A long time ago, there were prokaryotes, much like there are today. Some of the prokaryotes are prokaryotes with photosynthesis ability, which means they could make their own food using sunlight energy. Prokaryotes with the ability to use oxygen to produce ATP energy. Prokaryotes that are larger and could consume other prokaryotes. The endosymbiont theory is that some of these large cells engulfed some of these small bacteria, but instead of those small bacteria getting this digested, some of them remain intact and they begin to live as symbionts. This is believed to be ancestor of the heterotroph eukaryote cell. Eventually, some of the cell engulfed the small bacteria that could do photosynthesis in addition, and there was more living as symbionts. This is believed to be the ancestor of autotroph eukaryotes. Now we consider the bacteria that use oxygen to produce their own energy to have evolved to mitochondria, and the bacteria that use sunlight energy to produce their own food we consider to have evolved into chloroplast. It is likely that these ancient eukaryotes had an advantage in their endosymbiosis. It is also likely that the mitochondria developed first before chloroplast because nearly all eukaryote cells have mitochondria. It is just the eukaryote cell that can do photosynthesis can have both mitochondria and chloroplast. Fact that support endosymbiont theory. Mitochondria and chloroplast have their own DNA. Their DNA is arranged in a similar way to prokaryote DNA. The size of mitochondria and chloroplast tend to be similar to the size of bacteria. They divide in a way that is similar to how bacteria divide.
Prokaryote cell structure. The prokaryotic cell has several elements that allow it to function as a living organism. A prokaryotic cell is a type of cell that does not have a true nucleus or membrane-bound organism within the domains of bacteria and archaea are based on the prokaryotic cell, while all other forms of life are eukaryotic. Prokaryotic cell structure. Nucleus. A central region of the cell that contains its DNA. Ribosome. Ribosomes are responsible for protein synthesis. Cell wall. The cell wall provides structure and protection from the outside environment. Most bacteria have a rigid cell wall made from carbohydrates and proteins called peptidoglycans. Cell membrane. Every prokaryote has a cell membrane, also known as the plasma membrane, that separates the cell from the outside environment. Capsule. Some bacteria have a layer of carbohydrates that surround cell wall called the capsule. The capsule helps the bacterium attach to surfaces. Membrane. Membrane are thin, hair-like structures that help with cellular attachment. Pili are root-shaped structures involved in multiple roles, including attachment and DNA transfer. Flagella are thin, hair-like structures that assist in movement.
That's from our group. Thank you for listening.